Hello my dear students. Our new unit is reproduction, and our first topics in reproduction unit is cell cycle and mitosis. What is mitosis? You know, mitosis is the cell division. Actually, reproduction is one of the characteristics of life. You remember other characteristics, all the living organisms characteristics. They made up of cell. They need to obtain food, so they need nutrition. What else? Respiration to produce energy. Adaptation. DNA and RNA structure, or having nucleic acids. Excretion, get rid of waste. What else? All the living organisms have cellular structure. And the one is the reproduction, the ability of organism to produce one offspring. So this reproduction depends on cell division, based on cell division. In this picture you see the chromosomes, and in the different stages of mitosis. This is the mother cell, and this mother cell divides into two daughter cells, and this process starts from here and ends here, and this process is called mitosis. In this mitosis process, there are different stages, and the chromosomes are seen in different shapes. For example, in this part they are more fluid, but in this part they are more condensed. And in this part they are separated. And at the end of mitosis, one cell turns into two daughter cells. What are the differences of cell division for unicellular and multicellular organisms? For unicellular organisms like Omabia, Paramecium, bacteria, the cell division means reproduction, because one organism turns into two organisms. But for multicellular organisms like human, like animals, like plants, the cell division means development from a fertilized cell. What is fertilized cell? Fertilized cell is the zygote. It means growth, or it means repair. Cell division is an internal part of a cell cycle. What is cell cycle? Cell cycle is simply the life of a cell from formation to its own division. Cell cycle. You see in this picture, this is Amobia, and the Amobia divides into two. One Amobia divides into two, and it means reproduction for a unicellular organism. Cell division means reproduction. Actually, cell division for prokaryotes. What are the prokaryotes? Prokaryotes are bacteria and archaea. Prokaryotes, you remember, prokaryotes do not have nucleus, no nucleus. So, nucleus, if there is no nucleus in prokaryotes, they cannot divide by mitosis, because mitosis is the division of nucleus. So if prokaryotes do not have nucleus, therefore they divide by binary fission. What is binary fission? Binary fission is very simple, and it doesn't need spindle fiber formation. But one bacteria, for example, E. coli is one of the examples of a bacteria. E. coli can divide into two. First, the chromosome, bacterial chromosome, is doubled, copied, or replicated. The DNA amount goes into two. Then simply the cytoplasm, in this part, the cytoplasm is divided into two. This chromosomes is attached in the opposite sides of the cell membrane. Then at the end of the binary fission, two new bacteria or two new prokaryotes can be produced. It's very simple. It is not mitosis, because there is no nucleus in prokaryotes. Prokaryotic organisms divide by binary fission. and it's a reproduction for this organism. Not all the cells divide. For example, neuron cells in your brains, for example, neuron cells are non-dividing cells. But for example, other cells, bone marrow cells always divide to produce red blood cells. So division of the cell in a single organism also different. Some cells divide spontaneously, but some cells never divide in our body. Let's start the cell cycle. Cell cycle is the whole of an cell, and there is a DNA. We call the DNA material in the cell. We call this whole DNA material is genome. Genome is a new vocabulary. Genome, the whole DNA in the cell. Genome can consist of a single DNA molecule for prokaryotes, because remember the prokaryotic cells, they have single circular DNA inside the cytoplasm, but the genome for eukaryotic cell are different because they have cell membrane, they have nucleus, and they have many, for example, for human number of chromosomes, 46 chromosomes are packaged into the nucleus. You see in this picture, the blue ones are condensed in the condensed form of chromosomes. These are chromosomes. They are colorful, not always colorful, but they put some specific dye to see the chromosomes. And these pink parts are the spindle fiber to attachment to pull the chromosomes to the other side of the cell. First, I want to give you two new vocabulary. The first one is somatic cell. The second one is gametes. Somatic cells are the cells in your body except from reproductive cells. Reproductive cells are sperm or eggs in your body, and they are haploid. They have just one set of chromosomes. But in your somatic cells are actually body cells, all the body cells. For example, liver cells, lung cells, brain cells, all your body cells. And they are diploid, 2N, diploid. What does diploid mean? They have two sets of chromosomes. Eukaryotic chromosomes consist of chromatin, a complex of DNA and protein, and it condenses during cell division and turns to chromosome. Chromosome actually the condensed form of a chromatin. Before the cell division, DNA should be replicated. So replication or duplication of DNA occurs in the interphase, and then the chromosomes condense, and each duplicated chromosome has two sister chromatids. And these sister chromatids are attached to each other at the centromere regions. So these are new vocabulary. Chromatin, chromosome, sister chromatids, and centromere. I am going to explain this vocabulary on the picture. This is a unduplicated chromosome. I said it chromosome because it's more condensed. This chromosome is unduplicated. There is no DNA replication occurred yet. But after DNA synthesis, synthesis or replication of DNA, you are going to see the chromosome in this shape, and we are going to say it's a duplicated chromosome. In electron microscopy, you are going to see the duplicated chromosome in this figure. And one duplicated chromosome consists of two sister chromatids. This is one sister chromatid, and this one other sister chromatid. So the whole part is, this one is duplicated chromosome. One duplicated chromosome consists of two sister chromatids. Okay, where is the chromatin? We said that chromatin is composed of DNA plus protein. So this is the DNA, double helix structure of DNA, and these red ones are protein. Actually, it's histone protein. Histone protein. And this histone DNA is packaged into the histone protein to form a single fiber thin structure, and this thin structure is called chromatin. When the chromatin is more condensed, it turns into chromosome. This is chromosome. And this is duplicated chromosome because it's in X shape, and each chromosome consists of two sister chromatids. This is one sister chromatid, and this is another sister chromatid. And these chromatids are attached to each other in the centromere region. This is the centromere region. Also, this figure can explain it. This is simple DNA. This one, the first one. Second one is DNA with protein and folding in the proteins. DNA is folded into the protein. And this is the chromatin, fiber-like, thread-like structure. When the DNA replication occurs, the chromatin is seen in this figure. And when this chromatin is condensed, it turns into duplicated chromosome, and the two sister chromatids attached to each other on the duplicated chromosome in the center called centromere. It comes from center of a chromosome, centromere, center of a chromosome. The chromosomes number are one of the characteristics of each species. For example, prokaryotes only have one chromosome because prokaryotes have one circular DNA, and we call it just one chromosome. For example, crayfish have 200 chromosomes, but humans have 46 chromosomes in each somatic cell. These 46 chromosomes actually are 23 pairs of chromosomes. Diploid organisms, two set of chromosomes. Diploid organisms receive one chromosome from their female parent. We call it maternal chromosomes. And one chromosome comes from male parent. We call it paternal chromosome. During the fertilization, for example, this is your mother's egg, and this is your father's sperm. During fertilization, one set of chromosomes comes from egg, one set of chromosomes comes from sperm, and at the end of fertilization, 46 chromosomes occur. Actually, two sets of chromosomes. We are going to write in the chromosomal formula of human like that. 2n equals to 46 in the zygote and all body cells, all somatic cells. A matched pair of maternal and paternal chromosomes are called homologous chromosomes. Homologous chromosomes are very, very important vocabulary. You have to understand what is the differences between homologous chromosomes and what are differences between duplicated chromosomes. You see, there are two chromosomes here, chromosome 1, chromosome 2. And these two chromosomes, for example, this purple one comes from your mother and the green one comes from your father. And in your body, in your zygote, the two chromosomes are together. These two chromosomes, if these two chromosomes are responsible for the same characters, for example, eye color. If there is a gene for eye color in this chromosome and in that chromosome also for eye color, for example, it can be blue eye color from your mother, and it can be comes from your father, how can I say, green. Then you are going to have two genes for eye color, and in your phenotype it depends on which one is dominant to others. So these chromosomes, which are responsible for the same characteristics and which are similar in length and similar in structure, we call these chromosomes homologous chromosomes. Homologous chromosomes are actually, they are the pair of chromosomes, matched pair. After DNA replication, you are going to see the homologous chromosomes in this form. This one is unduplicated. This one is duplicated. Again, this is one chromosome, this is one chromosome, but the just only differences is this is unduplicated, this one is duplicated chromosome. When the chromosomes are duplicated, there are two sister chromatids. This is one sister chromatids, and this is another sister chromatids. These two chromosomes, still we call these homologous chromosomes. So if we define the homologous chromosomes are chromosomes pairs, one from each parent, maternal and paternal, that are similar in length, in gene position and centromere location. The position of the genes on each homologous chromosome is the same. However, genes may contain different alleles. For example, for eye color, it can be blue or green. This is a different allele for the same gene. These are the homologous chromosomes of all chromosomes of humans. This is called karyotype. In karyotype, this is the light microscopic photography of the human chromosomes, and you see in the first pair, you see they are same in the length. Second pair, third pair, there are 46 chromosomes, but actually there are 23 pair of homologous chromosomes. These two are sex chromosomes. We call them gonosomes. But other 44, these 44 are responsible for the body, responsible for other characteristics. These 44 chromosomes, we call them autosomal chromosomes. Autosomal chromosomes. But these two we call X and Y, or just for the female X and X, we call them sex chromosome or gonosomal chromosomes. centromere region. Centromere is the center of a chromosome. When the chromosome is duplicated chromosome, the sister chromatids are attached to each other on the centromere region. What is kinetochore? Kinetochore is composed of protein plus DNA, and in the centromere region of a chromosome, in this part, centromere region, this yellow structure are called kinetochore. Why the kinetochore is important? Because during the mitosis, spindle fibers, this yellow one, spindle fiber microtubules will attach to the kinetochores to pull these sister chromatids to the opposite side of the cell. They are going to pull this sister chromatid to this side and pull this sister chromatid to other side of the cell. Okay, what is cell cycle? Cycle is the whole life of a cell, and it composed of two main phases: mitotic phase and interphase. Actually, interphase is longer than mitotic phase, and in mitotic phase, DNA is condensed and you can see the chromosomes, but during the interphase, DNA is uncondensed and the DNA in the form of chromatin fiber. Interphase divided into three stages: G1, S, and G2. Simply in G1, cell grows. In S1, DNA replication occurs, the synthesis of DNA. And in G2, again the cell grows and it synthesizes important proteins for cell division. So we can say preparation for cell division. Preparation for cell division. The DNA replication only occurs in S phase, the synthesized phase of interphase. Let's look at the picture of cell cycle. This is the whole cell cycle, whole life of a cell. 90% of cells goes into the interphase. Interphase, this part. This whole part is interphase. In the first growth phase, cell grows and normal activities occurs. When the DNA is replication, it means cell should go to mitosis. If the DNA is replicated, then we can say that the cell will be going to divide into two. This is the S phase. G1 growth, S, DNA replication, and G2 growth and preparation for mitosis. They are going to duplicate the number of the centrioles and they are going to synthesize the important proteins for the cell division. And the second phase is mitotic phase. Mitotic phase composed of prophase, metaphase, anaphase, and telophase steps. And after the telophase, I want to write something, add something here. During the telophase, at the end of the telophase, actually, they are going to finish at the same time. There is one phase you are going to please add in this part. This phase is called cytokinesis. What is cytokinesis? Cytokinesis is division of cytoplasm. So, this fourth step simply called mitosis, but this last one is called cytokinesis. So mitotic phase divided into two, mitosis and cytokinesis, and mitosis is divided into four: prophase, metaphase, anaphase, and telophase. Yes, this is actually not mitosis, but mitotic phase. We can say mitotic phase composed of two major processes. The first one is the mitosis, nuclear division of nucleus. The second one is cytokinesis, division of cytoplasm. And mitosis have four phases: prophase, metaphase, anaphase, telophase. And at the by the late telophase, it occurs at the same time and finish at the same time, nearly the same time. It's the cytokinesis, division of cytoplasm. What happens in prophase? Three major events. Chromosomes condense. Actually, chromatin fibers condense and turns into chromosomes. Spindle fiber forms and chromosomes are captured by spindle fibers. What else? Chromosomes are duplicating during interphase. So we are going to see the chromosome in duplicated chromosome shape. And each chromosome will have two sister chromatids. And they are attached to each other in the centromere region. Spindle fibers are specialized microtubules, and spindle fibers are produced by centrioles. Centriole is an organelle responsible for spindle fiber formation and helps the division of cell. Centrioles occurs in pairs and duplicating during interphase, actually G2 phase of interphase, and each one pair of centrioles migrates to one pole of cell. Spindle fibers, when the spindle fibers fully form, nuclear envelope disappear, nucleolus disappear, and spindle fiber attach to the chromosome at their kinetochore structure, located on the centromere. Therefore, they organize the sister chromatids division into two. Let's look at the figure. What happens in prophase? Chromatin fibers converted into chromosome by condensing. You see, this is chromosome, this is chromatin, thin and very more thick. What happens? Centrioles duplicated and goes to each poles. Spindle fibers start to form between the centrioles. These spindle fibers attach to the kinetochore region of a chromosome. Nuclear envelope disappear, nucleolus disappear. These are the 6 main events occurring during the prophase. After prophase, you also see the different structures. Red things are the chromosomes of onion root, and in the animal cell, white fish, you see the chromosomes. During metaphase, just one event occurs. Chromosomes are lined up in the equatorial region of a cell, and each kinetochore facing each pole. You see, there are kinetochores in this part. Kinetochores face in each pole, and they are lined up in the equatorial region of. Yes, like this or this one. This is, we also the equatorial region in this figure, we are going to say this is metaphase plate. And the spindle fiber at the poles, we are going to say aster. In anaphase is actually separation of the sister chromatids. Spindle fibers attached to the kinetochores shorten and pull the sister chromatids to the other poles of the cells, and free spindle fibers lengthen and push poles of the cell apart. As occur, the duplicated chromosome separated each sister chromatid. This is one sister chromatid in duplicated chromosome separated, and each goes to each cell. After separation of sister chromatids, we are going to say these chromatids, now we are going to say these are chromosome again. Unduplicated chromosome. When the separation of, you see, the anaphase, they are separated, pulling by the spindle fiber. And at the telophase is the reverse of prophase. Again, spindle fibers disintegrate, disappear. Nuclear envelopes and nucleolus again appear. Chromosome revert extended phase state we called chromosomes converted into chromatin. thin structure again, and nucleolus reappear. You see the nuclear membrane start to reappear, nucleolus start to reappear. You see there are two nucleus almost formed, and when the nucleus is divided into two, cytoplasm also should divide, and we call this event cytokinesis. Cytokinesis is different in plant and animal cells. In animal cells, cleavage furrow occurs. Cleavage furrow is the pinching of plasma membrane by the help of microfilaments. To see, this is the pinching. By the help of microfilaments, the plasma membrane pinches, and you see the cleavage furrow. We call this cleavage furrow by the contracting of microfilaments occur and divided the cytoplasm divided into two. But in plant cell, because there is a cell wall, the pinching is impossible. Therefore, Golgi complex buds of some vesicle send some vesicle filled with carbohydrate vesicles and line up in equator, and they form a cell plate structure. And cell plate becomes new cell wall between two cells. You see, these are the vesicles come from the Golgi, and the vesicles contain some components of cell wall, and we call this structure cell plate. Cell plate. This is the difference between plant and animal. Cytokinesis in animal cell by cleavage furrow, cytokinesis in plant cell by cell plate formation. Cytokinesis does not happen all the time. For example, in muscle cells, mitosis occurs, but cytokinesis does not follow mitosis. So some cells may have more than one nucleus because it increases the number of nucleus, but the cytokinesis. In multicellular organisms, mitotic cell division is used for growth, development, repair of the body tissues. And at the end of the mitosis, different from the meiosis, the two daughter cells are identical to the mother cell, and chromosome numbers does not change. There is no genetic variation in the mitosis. You see in this picture the differences in microscopic photographs, mitosis in plant and in animal cell. The most important difference is the cell plate formation at the end of telophase. This is the summary of mitotic phases and prophase and prometaphase. The differences between prophase and prometaphase. Actually, the two are prophases. Prometaphase is the last part of prophase. In picture, it's the microscopic picture of onion of root cells, and in this picture you can differentiate the metaphase phases by the metaphase plate, anaphase by the separation of sister chromatids, telophase, you see two different nucleus start the formation, and prometaphase and prophase, and these are interphases. Number of the chromosomes during the G1, S and G2, during the interphase part, number of the chromosome does not change. In anaphase, the number of the chromosomes becomes double. But if you look at the DNA amount, because DNA synthesis occurs during the S phase of interphase, the DNA amount is duplicated during the S phase, and again it's become half when the cytokinesis occurs.